So the pigment like the melanin can be eliminated by the enzyme action in our body. Question number 2. The complete active enzymes consisting of both protein and a cofactor is called as. Options are option A, isoenzymes, option B, holoenzymes, option C, apoenzymes, option D, none of this. And the correct answer is option B, holoenzymes. Now, enzymes are normally proteinaceous in nature and they consist of two parts. One is the active form and second is the inactive component. So, the inactive component of an enzyme is called as apoenzyme or apoprotein. So when it is combined with a cofactor, which can either be a metal like magnesium or iron, or it can be any organic molecule such as coenzymes like NAD plus, etc. So when this cofactor is combined with the inactive part of the protein, together it is called as the holoenzyme. The other options such as the isoenzyme, these are the enzyme that differ in the amino acid sequence, but they catalyze the same biochemical reaction. The apoenzyme, as I said earlier, is nothing but an inactive protein part of an enzyme. So here our correct option is holoenzyme. Question number 3. Which enzyme is the major proteolytic enzyme of the fourth stomach of the suckling calf? Options are option A, papain, option B, allicin, option C, renin, option D, all of the options. And the correct answer is option C, renin. So, renin is an enzyme which is present in the fourth stomach of milk-fed ruminant animals such as the goat, lamb, or the calves. So, the fourth stomach is also called as ebosum, and this specific enzyme, the renin enzyme, is present only in the milk-fed uh, animals. So it is normally obtained from the calf. So this enzyme normally converts the protein part of the milk, which is the casein, into the simpler uh, casein compounds. So the other enzymes like the papain enzyme, it is also an proteolytic enzyme, but it is present in plants such as the papaya. Allicin is not an enzyme, but it is a compound which is present in garlic. The enzyme here is the allinase enzyme, which converts alanine into allicin. So here our correct option is renin enzyme. Question number 4. Bacteria producing proteolytic enzymes are option A, Salmonella, option B, Arvina keratopora, option C, Shigella dysenteria, option D, Clostridium perfringens. And the correct answer is option A, Salmonella. Now the other options like the Arvina keratopora, it is a bacteria that causes bacterial soft rot, which is a very common disease observed especially during the storage of fruits and vegetables. During the rainy season, it is especially observed in the onions. The Shigella dysenteria is a rod-shaped bacteria that causes shigellosis, a disease that includes the symptoms such as diarrhea, fever, and abdominal pain. The Clostridium perfringens is an gram-positive anaerobic spore-forming pathogenic bacteria, which is very common, and it is normally it causes food poisoning. So here the correct option is Salmonella, which produce proteolytic enzymes. Question number 5. Enzymes which convert starch to maltoses. Options are option A, maltase, option B, diastase, option C, invertase, option D, hydrogenase. And the correct answer is option B, diastase. Diastase is an alpha amylase enzyme which normally converts the complex molecules of carbohydrates such as starch and other glycogen or glucan molecules to simpler sugars such as maltose or other disaccharides or monomer units. The enzyme the maltase normally converts maltose sugar into glucose. The hydrogenase enzyme catalyzes the conversion of hydrogen molecule to proton and electron by heterolytic splitting mechanism. The invertase is an enzyme which causes hydrolysis of sucrose, which is a disaccharide, into its monomer unit, namely the glucose and the fructose. Question number 6. Which of the following enzymes uses urea as a substrate? Options are uricase, option B urease, option C amylase, and option D catalase. And the correct answer is option B urease. Urease is an enzyme that catalyzes the hydrolysis of urea into ammonia and carbon dioxide. These byproducts, especially the ammonia, is taken up by the soil microbes and plants, which is then utilized as nitrogen fertilizer. The other enzymes such as the amylase is an enzyme, or we can also call it as a special protein that helps to digest the carbohydrates. It is generally present in the pancreas and salivary glands of human body. The catalase is an enzyme. It is also called as antioxidant enzyme that breaks down the hydrogen peroxide to water molecule and molecular oxygen. It is present in all living organisms those are exposed to oxygen. Uricase is not an enzyme. Question number 7. Which of the following has catalytic activity? Options are option A collagen, option B nucleosides, option C glycogen, option D RNA. And the correct answer is option D RNA. RNA means ribonucleic acid. Now the ribosomes are the nucleic or the ribonucleic acid enzymes that have the ability to catalyze the specific biochemical reaction, including the splicing of the RNA into simpler genes. Now RNA is a molecule which is present in most of the living organisms and viruses. It is made up of the nucleosides. It has a structural similarity to DNA, but DNA has two strands or the double helical structure, while RNA has single strand. The other options such as the nucleosides are the ribose sugar, which are attached to the nitrogenous bases and phosphatase or the phosphate groups. Collagen is the most abundant protein inside the human body, and it acts as a building block for our skin, muscle, bones, ligaments, and other connective tissue. Glycogen is a stored form of glucose which acts as a main source of energy. Question number 8. Choose the correct enzyme substrate pair. Options are option A renin casein, option B protein amylase, option C carbohydrate and lipase, option D lactose maltase. Correct answer is option A renin casein. Now casein is a protein which is present in milk and it requires an enzyme. So renin is an protein digesting enzyme which curdles the milk by transforming the caseinogen obtained from the casein into the soluble compounds. If we see the other options like the protein and amylase, herein the protein requires proteolytic enzymes to be broken down. Amylase is an enzyme which requires carbohydrate or starch compounds to be broken down. Option C, where the carbohydrate is present, it requires amylase like enzyme to be broken down into simpler sugars. Lipase is an enzyme which requires lipids or the fats as a substrate. In option D, we have lactose as a substrate. Uh, on the lactose, which is an disaccharide present in the milk, it requires lactase enzyme to broken down into simpler sugars. Maltase is an enzyme which requires maltose as the substrate sugar. Question number 9. In a cell, digestive enzymes mostly occur in options are option A mitochondria, option B ribosomes, option C lysosomes, option D plastids. And the correct answer is option C lysosomes. Lysosomes are formed by the Golgi bodies. These are the membrane bound vesicles which contain the digestive and hydrolytic enzymes such as the glycosidase, protease, and sulfonates or the sulfates. The mitochondria is also known as the powerhouse of the cell, and it helps in the formation of energy in the form of ATP. The ribosome help in the synthesis of the protein, and the plastids have chlorophyll which help in the photosynthesis. So here the correct option is lysosomes. Question number 10. Diastase takes part in the digestion of options are option A starch, option B amino acid, option C protein, option D fat. Correct answer is option A starch. Diastase is a group of enzymes that catalyzes the breakdown of starch into maltose. It is also called as alpha amylase, which breaks down the starch into simpler sugars like maltose. The other components like the amino acid and protein requires protease enzyme to break down it into simpler molecules. The fat is broken down by an enzyme called as lipase. Question number 11. The enzymes which converts glucose to glucose 6-phosphatases. Options are option A enolase, option B aldolase, option C hexokinase and option D fumarase. And the correct answer is option C hexokinase. It is an enzyme that converts glucose to glucose 6-phosphatase and then traps the glucose molecule inside the cell, and it is then further goes for the process of glycolysis. Aldolase is an enzyme. It is also called as cytoplasmic enzyme, which is involved in the glucose and fructose metabolism. Fumarase is an enzyme that converts fumaric acid into malic acid. Enolase is also called as phosphopyruvate hydrate enzyme, which converts phosphoglycerate into phosphoenol pyruvate and water, which is a second step in the glycolysis. Question number 12. Which enzyme can digest plant proteins? Options are option A renin, option B pepsin, option C erepsin, option D all of the above. And the correct answer is option B pepsin. Here all the options where all the enzymes are given are the proteolytic enzyme. So the renin is an enzyme which normally is utilized to digest the milk proteins, especially the caseinogen, into smaller component of casein. The pepsin is an enzyme that converts protein into smaller peptide bonds, and thus it helps in digesting the plant proteins which are normally taken up from the food that we ingest. Erepsin is an enzyme that digests the peptones into amino acid, and it is secreted by the intestinal glands and pancreas. Question number 13. Hydrolysis of starch occurs with the help of options are option A peptidase, option B sucrase, option C lipase, option D amylase. And the correct answer is option D amylase. Now hydrolysis of starch involves addition of water molecule and breaking down at glycosidic bonds between glucose molecule, which are then further formed into smaller particles of polysaccharides, disaccharides and other glucose units. The enzyme which is used in this hydrolysis is the amylase enzyme. It can be alpha amylase or beta amylase. The other enzymes such as the peptidase enzyme, it is protein based enzyme which breaks down the long chain of protein with the help of water at peptide bonds, and it converts into smaller units of proteins. Lipase is an enzyme which break down the lipids or the fats into triglycerides and fatty acid molecules. Sucrase is an enzyme which break down the sucrose, which is a disaccharide, into monomer units, namely the glucose and the fructose. Question number 14. Esterases belong to the category of option are option A lyases, option B synthetases, option C hydrolases, option D transferases. And the correct answer is option C hydrolases. Now the esterases are the enzyme which hydrolyze the ester, amide or thioester bonds which cause to produce the prodrug activation or detoxification. The other enzymes such as the lyases, these are the enzymes which are responsible for addition or elimination of bond into a reaction. For example, it can break the carbon atom and add another atom such as the oxygen or sulfur in its place. The synthetases are the enzyme. These are also called as lyases enzyme, and they catalyze or join the two molecules by forming a new chemical bond, typically by hydrolysis. Now transferase are the enzyme which catalyze the transfer of specific group of uh, functional group from a donor to an acceptor. Question number 15. Which enzyme bring about the cleavage of specific covalent bond and removal of groups without hydrolysis? Options are option A lysases, option B ligases, option C hydrolases, option D transferases. Correct answer is option A lysases. The other enzymes such as ligases, these are the enzymes that are used to join the DNA strands. The hydrolases are the enzyme that catalyze the bond cleavage with a reaction with water. And transferase are the enzyme that catalyze the transfer of specific functional group from a donor to an acceptor. Question number 16. Enzyme that catalyze rearrangement of atomic grouping without altering molecular weight or number of atoms is option A lysases, option B ligases, option C isomerase, option D oxidoreductase. And the correct answer is option C isomerase. These are the enzymes that catalyze the rearrangement without altering the molecular weight or the number, and thus it is called as isomerase. The other enzymes such as the lyases, these are the enzymes that bring about the cleavage of specific covalent bond and removal of groups without hydrolysis. Ligases are the enzyme that joins the strands of DNA, and the oxidoreductase enzyme catalyze the transfer of electron from a donor, which is also called as a reductant, to an acceptor, which is the oxidant. Question number 17. Amylase is an example of. Options are option A oxidoreductase, option B transferase, option C hydrolase, option D ligase. And the correct answer is option C hydrolase. Amylase are a group of enzymes that hydrolyze the starch by breaking the glycosidic linkage, that is the 1,4 linkage, and it is also called as the glycoside hydrolase enzyme. So the hydrolase is the correct answer. The oxidoreductase is a group of enzymes that catalyze the transfer of electron from a reductant or a donor to an acceptor or an oxidant. Now, transferase is an enzyme that transfers the specific functional group like the methyl or the glycosyl group from a donor to an acceptor. Now, ligase is an enzyme that joins the strands of DNA. Question number 18. Lipase breaks the. Options are ester bond, option B peptide bond, option C hydrogen bond, option D glycosidic bond. And the correct answer is option A ester bond. Now lipase are the hydrogen soluble or the water soluble enzymes that hydrolyze the ester bonds of water soluble substrates, for example the triglycerides. They further break down the triglycerides into diglycerides or the monoglycerides and the fatty acid. The peptide bond is broken by the protease enzyme, whereas the hydrogen bond is broken down by the helicase enzyme. The glycosidic bond is broken down by the glycoside hydrolase enzyme. It is also called as glycosidase enzyme. Question number 19. Genetic engineering requires enzymes. Options are option A DNase, option B amylase, option C restriction endonuclease, option D lipase. Correct answer is option C restriction endonuclease. In the genetic engineering, the enzymes are very specific in nature and only those are employed which can break or join the DNA strands. So in genetic engineering, whether it is an RNA or DNA, we require very specific enzymes. So the restriction enzymes are used for this purpose. The other enzymes like the amylase, it is a starch breaking enzyme, and lipase is a lipid breaking enzyme. So correct answer is the restriction endonuclease. Question number 20. Restriction endonuclease is employed for. Options are option A joining strands of DNA, option B cutting RNA, option C cutting single strand DNA, option D cutting double strand DNA. And the correct answer is option D cutting double strand DNA. Restriction endonuclease is also known as restriction enzymes, which are specific in nature and they are specifically employed in cutting of the double strand DNA. These are the enzymes which are isolated from various strands of bacteria that recognize and cleave the specific double-stranded DNA sequence, which is also called as restriction sites, with the majority of sites consisting of the 4 to 7 nucleotides. Now the joining of strands of DNA, it is done by enzyme which is also called as ligase. Ligase is a joining enzyme. The cutting of the RNA is done by spliceosome. Question number 21. The fastest enzyme is options are option A pepsin, option B carbonic anhydrase, option C DNA gyrase, option D DNA polymerase. And the correct answer is option B carbonic anhydrase. Now carbonic anhydrase is an enzyme that is present or found in red blood cells, gastric mucosa, pancreatic cells, which normally converts the carbon dioxide and water into carbonic acid, protons, and bicarbonate ions. It catalyzes the reversible reaction between carbon dioxide and water to form the carbonic acid. It also has another function, that is the formation of hydrochloric acid by the stomach. Question number 22. Fat is hydrolyzed by the enzyme known as, options are option A trypsin, option B lipase, option C pepsin, option D amylase. And the correct answer is option B lipase. Now lipase is an enzyme which breaks down the fats or the lipids into triglycerides and fatty acids when hydrolyzed. The enzyme trypsin is an protein digesting enzyme which is secreted by the pancreas. It is also present in the small intestine. Pepsin is an enzyme which is present in the stomach and it is activated at lower pH, so that the stomach acid, which is the hydrochloric acid, has very low pH. Thus it gets activated in the stomach and it digests the protein from the food. Amylase is an enzyme which digests or which converts the starch into simpler sugars. Question number 23. The term apoenzyme is applicable to, options are option A simple enzyme, option B protein part of conjugate enzymes, option C organic cofactor of conjugate enzymes, and option D the inorganic cofactor of conjugate enzymes. And the correct answer is option B the protein part of the conjugate enzyme. Apoenzyme is also called as apoprotein, which is an enzymatically inactive protein part of an enzyme which requires a cofactor for its activity. The cofactor can be any metal like the magnesium or the iron, or it can be any organic molecule. The organic molecule is also called as coenzyme, such as the NAD+, NADP+, or FAD2+. Now the cofactor which is tightly bound to the apoenzyme is called as the prosthetic group. For example, the heme prosthetic group which is attached to catalase or the peroxidase enzyme. Now the apoenzyme becomes enzymatically active only when it is bound to a cofactor, and together it is called as haloenzyme. So here the correct option is the protein part of the conjugate enzyme. Question number 24. Zymogen is, options are, it is an enzyme poison or a modulator, enzyme precursor, or enzyme inhibitor. Correct answer is option C, enzyme precursor. Now enzyme precursors are the simple proteins that become catalytically active by the hydrolysis process. These precursors are also called as zymogens or proenzymes, and these are normally present in the digestive tract of the humans. These are required to convert the inactive substance into active enzymes. Now, the enzyme inhibitor is a substance that blocks the action of the enzyme. Normally, the enzymes are required to speed up the chemical reaction in the body, and it takes part in the cell function. For example, in cancer treatment, if the cell is growing through a particular enzyme, we then introduce an enzyme inhibitor that will block the certain type of enzyme which will promote the cancer cell growth. Now, enzyme modulator is a drug which modulates the enzyme activity either by increasing or reducing its activity. Thus, enzyme modulator can be both. It can act as either the activators if it increases the activity, or it can also be called as inhibitor if it decreases the activity. Depending upon if we want to increase or decrease, the enzyme modulators are used. Question number 25. Enzymes used in chill proofing of beers. Options are option A ligase, option B protease, option C pectinase, and option D amylase. And the correct answer is option B protease. Chill-proofing is a process where beer is cooled to sub-zero temperatures or almost to 0 degrees Celsius for a longer period of time, so that the proteins and the polyphenols which come from the malt in the beer coagulates to form larger particles which can be then filtered to produce clear and haze-free beer. To reduce the time taken to break down the proteins, the protease enzyme is used. The other enzymes like the pectinase is used for the breaking of pectin which comes from the fruits and vegetables. Amylase enzyme is used to break down the starch into glucose or other monomer, monomer unit of the sugars. And the ligase is an enzyme which is used to join the restriction endonuclease fragments in the structural or the DNA engineering. With this, we have completed all the 25 questions, and let me make a quick appeal to you all. If you have still not installed our app, FoodTech Online, please do so. It will help you prepare for the entrance exam related to food technology. So the link is given already in the description box. It covers the major topics like the food microbiology, the food engineering, and other subjects too. It will prepare you for the other exams like the GATE exam, the NET exam, etc. So please go through this. Thank you. Also, please subscribe to our YouTube channel and also join us on the social media via the link given in the description.